So welcome to this briefing session on precision breeding. Last year, primary legislation was brought in the form of the New Genetic Technologies Precision Breeding Act, and this essentially set the definition of what a precision-bred organism is, and that's one where genetic technologies have been used to produce an end product that could equally have been produced using traditional breeding approaches. And so it set out to have a science-based and proportionate regulatory system, and therefore removing these products from the GMO legislation. It moves us in line with other countries who are also moving in that direction of regulating precision-bred organisms more in line with traditional breeding approaches. And ahead of this legislation going through, so in April 2022, the rules were relaxed with regards to field trials. So while in the field evaluation stage, these products or these crops are known as qualifying higher plants. And since the rules were relaxed, over 16 qualifying higher plant field trials have taken place. And this puts us ahead of other countries that haven't yet changed their rules. So it puts us ahead of Europe, for example, where these products would still be, these crops would still be considered GMOs. And as a result, we've seen a big increase in interest from industry and investors into these technologies. So we're now at the secondary legislation stage. So that is the changes to legislation that need to be put in place in order to implement the new Genetic Technologies Precision Breeding Act so that we can bring products to market. And so the aim of this briefing session is to bring some of our newer MPs up to speed with the science behind precision breeding and to give an idea of some of the applications that we can use these new technologies for. Now, before I move on to the science, I thought I'd give you a quick introduction to the Norwich Research Park for those of you who don't know us. So we're based in South Norwich, and we are home to 4 research institutes. So the John Innes Centre, which is a plant and microbial research centre, the Sainsbury Laboratory, which has a focus on plant health, the Earlham Institute, which is a genomics and synthetic biology sort of research facility, Quadram Institute, which has a focus on food and how it affects our health. We're particularly interested in the gut microbiome. And we're also home to the University of East Anglia and the Norfolk and Norwich University Hospital. And also we are home to over 30 companies as well, so more than 30 biotech companies on the research park. And a lot of those companies have resulted from spin-outs from our own research, but also spin-ins and companies relocating to the Norwich Research Park to make use of our facilities and our expertise. And of perhaps particular interest to this briefing session, we have Tropic Bioscience. So they're one of our larger companies on the research park. They employ over 150 people. They've been here for, I think, 6 years. I probably got that wrong. And they initially came as a spin-in, so a small company, a handful of people, and they've grown considerably over that time. So that's a huge amount of investment that's come into the region as a result of the region's expertise. And they have an interest in gene editing bananas, coffee and rice. We've got a much smaller company who's just joined the Norwich Research Park called Alora, and they're focused on gene editing in rice as well. So we are beginning to see sort of investments coming into the region as a result of changes in legislation. And as a result of these changes to legislation, we've seen increased interest from industry. So together, the John Innes Centre with British Sugar and the British Beet Research Organisation, so BBRO, and Tropic Bioscience, we have funding with British Sugar to develop a transformation or a precision breeding pipeline for sugar beet. And the aim of that is to produce, using genetic technologies, sugar beet that is resistant to virus yellow disease. And that has a significant impact on the sugar beet industry in the UK in terms of lost yield. And the reason why that's such a big problem is because we've got less chemistry to spray to get rid of aphids, and these are the transmitters of this virus. So that's a £1 million investment into the region as a result of that collaboration. And you may also be interested in the fact that we've had over £300 million worth of investment from the government in order to develop new buildings and infrastructure for the John Innes Centre and the Sainsbury Laboratory. So this is being referred to currently at the moment as the HP3 project. So that's Healthy Plants, Healthy People, Healthy Planet. And in total, this will be over £350 million investment into new buildings and infrastructure. And that's because if we're going to continue doing the world-leading research that we are doing, we need better facilities and infrastructure. And we hope that that investment will also attract more funding to the region in terms of scientific collaborations and better research outputs. Also on the Research Park, the Anglian Innovation Partnership, who are the company that oversee the Norwich Research Park, they also have plans for new buildings totalling over £200 million. So there's huge investment on the Norwich Research Park into buildings and facilities. And the Norwich Research Park has a policy advisory group. So this brings representatives from each of the 4 institutes together with the university, the hospital and Tropic Bioscience. And collectively, we provide a voice to engage with policymakers in sessions such as today to provide science-based evidence to help inform policy changes. So in regards to the new Genetic Technologies Precision Breeding Act as it went through Parliament, we have done a lot of work over the last couple of years working with DEFRA and the FSA during both their consultation phase, but also as they were drafting the bill, going out briefing MPs, explaining the science and the potential applications, as well as our scientists providing evidence at the select committee stage as this act passed through Parliament, and as well as attending various APPG science and technology in agriculture events, and going out and talking to industry as well as to the science, but also explaining what changes to legislation are likely to mean for them as well. So, as I say, the purpose of today's session is to bring you guys up to speed to explain the science and the potential applications of these technologies. So just to recap on a couple of terms that might be useful. So DNA, this is the information needed to make and maintain us, and our DNA is made up of these building blocks of chemical bases, which we refer to in this cartoon with the letters A, C's, T's and G's. So you can almost think of these as the alphabet of life. We all originally came from the same evolutionary thing. So the A, C's, T's and G's that are in you are exactly the same as the ones you'd find in a wheat plant, for example. When you hear reference to genetic sequence or genomic sequence, it means we now have the technology which allows us to read the order in which these letters appear throughout our genome. And something like the wheat genome will contain 17 billion bases or letters. And genetic variation is, if we were to compare the genome sequence between species, we'll see that there's a different order of letters, and that's genetic variation. And there was a really interesting study where 400 genomes of tomatoes were compared, and they found that some had 33,000 genes and some had 36,000 genes. So you can take away 3,000 genes and it's still a tomato. But most sort of genetic variation is much more subtle changes to that. So it can just be deletion of a few bases or just the changeover in that order in which bases occur. So that genetic variation can be a result of natural mutations. It's the basis of all evolution. And this is what plant breeders have been looking at for centuries. So they look for mutations or changes in that order of letter that gave rise to different traits. So for example, something like these different vegetable types, they're all the same species, but different traits that have been selected for over time have given us bigger leaves, to give us kale, or denser flower bud formation, to give us things like broccoli. And a plant breeder can only select from the genetic variation that's available to them. So it's very much a case of sort of hunting and looking for it. And so in the 1930s, a method called mutation breeding was introduced as a way of speeding up the occurrence of these mutations or genetic variation. So what's happening at the molecular level is you can lose sections of DNA, or you can create a break in the DNA, and as the plant attempts to repair itself, it may delete the odd base or recruit the wrong letter. And so you've introduced a point mutation. And then a breeder would look to see whether new desirable traits were found, and then he would breed those into his breeding lines. The problem with mutation breeding is you're introducing hundreds of thousands of random mutations into your lines, and there's no control of where they would be found. Whereas technologies that we have now, so we have access to all this genomic information. So if you think that plant breeding till now has kind of gone on without actual any knowledge of DNA and genetic sequence. So we now have access to all that information. And we have technologies, so genetic technologies, things like GM or gene editing, which now allows us to go in and make very specific changes to an organism's DNA. And just to sort of perhaps relate bases to genes, a given gene can be anywhere between a few hundred bases long, or it could be a couple of million bases long. So genetic technologies. So gene editing is an example of new genetic technologies. There are a number of different gene editing tools out there, but one called CRISPR-Cas is probably the most popular. That stands for clustered regularly interspersed palindromic repeats, which doesn't help explain the technology, but it was first identified in bacteria. And it was first used to edit genes in plants in 2013, and we've been routinely using it now at the John Innes Centre to edit the genomes of brassica crops, wheat crops, barley crops, potato and tomatoes. And so you can think of this as a tool which allows us to make cuts to the DNA at very precise locations. So we now have genomic sequence for most species that we work with. So we can now direct where we make breaks in the DNA. So we can delete sections of DNA, or we can make a break to the DNA. We hope that the plant attempts to repair that break, and in doing so, it could delete or insert the wrong base. So we're not adding anything new, we're just making deletions or cuts to the DNA. And we would refer to these as gene knockouts because these small edits are often enough to stop that particular gene from functioning. And these would be regulated as precision-bred organisms under the new Genetic Technologies Precision Breeding Act, because the endpoint could have been achieved through traditional breeding approaches, but we've just gone there in a much more direct route. Now we can also use this technology to open up the genome at a precise location in order to insert or replace genetic sequence, and we would refer to these as knock-ins. These would continue to be regulated as GMOs if the gene was from another species. So if we were to take a disease resistance gene from a wheat plant and put it into tomato, they would count as transgenic. So they're going from one species to another, and they would need an additional regulatory oversight. But if we were to take a gene from, say, a potato plant and reintroduce it into a potato plant, this would be referred to as cisgenic, because the endpoint could have been achieved through traditional breeding approaches. And in the picture we've got below, this is an example of blight-resistant potatoes. So you can see the nice green plants. So these contain 3 genes from wild potato relatives that have been introduced into commercial varieties in order to withstand late potato blight. So this is a really nice example of where we can use biological solutions to tackle these sorts of diseases rather than rely on fungicides. But I'm going to focus more on the gene knockouts because this is the majority of crops that are coming to market or nearing commercialisation will be where we've made deletions or small edits to the plant's existing DNA and where we've not introduced new genetic sequence. So this is a worked example of where we've used gene editing to create a gene knockout in order to identify or confirm that that particular sequence of DNA controls that particular trait. So what I'm showing you here is a field of oilseed rape. Now our farmers face some great challenges in even getting to this stage, but what a farmer doesn't want to do is lose some of the seed at the end of that as a result of premature pod shatter. So can we use gene editing to edit the genes responsible for pod formation in order to stop those pods shattering prematurely in the field and losing yield? So in this rather frightening looking slide, what I'm showing you is an example of genomic sequence. If I was to show you the entire genome sequence for brassica, I would need to share with you several million screenshots like this, so vast amounts of genetic information that we have available to us. And by comparing genomes from different species where we have knowledge of which genes control which traits, we can predict which gene sequence or which sequence of DNA we believe controls that trait in this crop. So that becomes our candidate gene. This is all done on a computer at this stage. So we find the sequence that we believe controls that trait. We would then look to identify a 20 base sequence that is unique to just your gene and nowhere else within the genome. And then we'd design a molecular guide which we'd introduce into the cell, which would hunt out and search for that genetic sequence. And you can think of this a bit like a Google search engine. And once it finds that sequence, it locks on. And then we introduce a nuclease or a Cas protein, which cuts the DNA at that location. So what we then hope is that the plant will attempt to repair itself, and in doing so will introduce an error. And this is what it looks like in reality. So here you can see our original genetic sequence at the top, where we've designed our guide, and the circle shows where we've made that break. And you can see that the sequence has changed in the edited line, so a C has been replaced with a T, and we've lost a G base. Now the image that I'm showing you is a cross-section of a fruit pod, and where the green arrow is showing is where that valve margin forms, so that's at the point at which those pods would break open. And in the edited plant, you can see that valve margin no longer forms, and so those pods would need to be broken mechanically in order to release the seed. Now for us, this wasn't a commercial endpoint for this particular project. It was really for us to use these technologies to confirm this is what that gene does. So because although we've got access to this genomic sequence, we don't know what everything does in that. So these technologies are really powerful research tools for us to help us understand what genes control which traits. So as well as being a really powerful research tool for us to help us understand the function of genes, we're also seeing gene editing has huge potential to actually make benefits of the science that we're unraveling in order to make huge benefits to help farmers and actually across the whole supply chain. So we're seeing work to develop more resilient crops. So there is work to develop pest resistance and disease resistant crops. It's an example here of yellow rust resistance in wheat. So two small deletions have made that plant less susceptible to yellow rust infection. We are seeing the use of gene editing to develop new or perhaps underutilized crops. So can we use this as an ability to perhaps domesticate new crops for UK markets? Can we use gene editing to adapt plants to climate change, so producing drought-tolerant crops as well? Can we increase yields, so either by increasing grain size or reducing yield losses? So there is now a pod shatter resistant oilseed rape that is in field trials. Can we use it to perhaps delay flowering in broccoli so that it extends the harvest period? And can we create healthier foods as a result of gene editing? So already on the market, we have this soybean with healthier oils with no trans fats, and we've got salad greens with higher nutrient levels. And at the John Lennon Centre, Kathy Martin's group have developed this vitamin D tomato. So something like 40% of the UK's population is deficient in vitamin D, and this offers a plant-based solution to that. So eating 2 of these tomatoes would be enough for you to get your vitamin D intake for the day. So it's very much moving us into this era of very much intelligent and precision-based breeding. And this is going to have great benefits as we try and tackle these sort of global challenges of feeding more people, but also giving them more healthier foods as well. And as we move towards better, more sustainable farming and a move to net zero. So just to sum up, we face these global challenges of producing more food, more sustainably to feed a growing population in a changing climate. And precision breeding has the potential to help us achieve these goals, along with a vast array of traditional breeding approaches as well. So it's important to remember that these new genetic technologies are tools. So depending on how you use them will depend on how they're regulated. So gene editing doesn't automatically mean a PBO. A precision-bred organism will only be those that have used new genetic technologies to reach an endpoint that could have been achieved through traditional breeding approaches. But it recognises that these are new technologies, and so some form of additional oversight is necessary. These products, along with traditional breeding, still will go through several years of field evaluations to ensure that we're bringing safe products to market, though safety isn't being compromised here. GMOs will be those where we've used these new genetic technologies to introduce new genetic sequence from a different species, and so it couldn't have been achieved through traditional breeding approaches. So these products will still require that additional regulatory oversight of GMO legislation in order to ensure that safe foods are brought to the market. And I'm going to leave it there. Very happy to follow up with any Q&As afterwards.